So one of the key areas of research for Shuki and his collaborators is biological and molecular systems. And that will be the first of our research panels. And please welcome Eric Winfrey, Mark Riedel, Ho-Lin Chen, and Damian Woods.
Hello, everyone. For those who don't know me, I'm Eric Winfrey. I have the honor of chairing the first panel discussion today. Our subject is biological and molecular systems. The format of this panel, and I think most of the ones later in the day too, will be that I and the other panelists will begin by offering 3 minutes of thoughts each, which should get us about halfway through our allotted time. And then we'll launch into a Q&A part. So please be ready with questions, observations, memories, or whatever else you have up your sleeve. I'll start by introducing the panelists. Please raise your hand or your flower petal when I call your name. So Andrei Levchenko is professor of systems biology at Yale. He did postdoctoral work at Caltech from 1998 through 2001, where he collaborated with Shuki and Paul Sternberg on modeling scaffold proteins and signal transduction, among other things. Sadly, due to a series of flight cancellations and other problems, he was not able to join us today. But I will present his slides with his notes for him. Mark Riedel is professor of electrical and computer engineering at the University of Minnesota. He did his PhD with Shuki on cyclic combinational circuits, graduating in 2003, where he was awarded the Wiltz Prize for Best Thesis in Electrical Engineering at Caltech. In 2009, he was one of the founding organizers of the International Workshop on Biological Design Automation, which has been held yearly since then. So glad you could join us for this panel, Mark. Ho-Lin Chen is professor of electrical engineering at National Taiwan University. He did his postdoc in my group from 2007 to 2011, during which time he interacted with Shuki and Shuki's student Hong Chao Zhou, who unfortunately was not able to travel from China to make it here today. Ho-Lin has been a pioneer of the mathematical analysis of computation in stochastic and deterministic chemical reaction networks. Hope you're not too jet-lagged, Ho-Lin. Damian Woods is a professor of computer science at the Hamilton Institute, Maynooth. He was the Molecular Programming Project postdoctoral fellow from 2009 to 2016, spending time with Shuki's group and with my group. His research at Maynooth is notable for having both very theoretical dry lab theory and very experimental wet lab experiments. Thank you for joining us, Damian. Okay, so I'm going to now start with my 3 minutes. Shuki began his professorship at Caltech in 1994 when I was a third-year graduate student. I took his class CNS 188 in winter of 1995. The course, entitled Computation Theory and Neural Networks, introduced me to Shuki's complexity theory for Boolean functions, computed with polynomial threshold circuits, harmonic analysis. It was astounding to me that one could prove rigorously that neural network architectures can be exponentially more efficient for computing certain functions than the traditional and or not circuits, whole classes of functions. After I started my own group in 2000, Shuki had a formative influence on the establishment of my group's intellectual culture due to a number of people who sort of migrated back and forth between our groups during a critical period. The first was Matt Cook, one of my best friends since high school, who came to do his PhD with Shuki. No surprise that whenever Matt got tired of thinking hard, he'd wander downstairs to our lab and listen to what was puzzling us, and there on the spot transform it into a beautiful insight. Then there was my student David Soloveichik, whose search for everything fundamental led him to Shuki and to Matt. and to a pair of wonderful papers on chemical computation. Ho-Lin and Damien, who you'll hear from in a minute, were also part of that vibrant circulation, as was Lulu, whom you heard from earlier. Shuki, being my upstairs neighbor, was undoubtedly one of the best things to happen to me at Caltech. I'd like to conclude by mentioning how theory can inspire experiment. The topic is the Hopfield associative memory, a neural network model where neuron Xi receives input from neuron Xj via weight Wij. Hopfield explained how recalling a memory pattern corresponds to minimizing a quadratic energy. And the proof he and everyone else used to show this involves some straightforward algebraic manipulations. Shuki looked at it from another perspective. Instead of neurons changing color to indicate whether they were on or off, Shuki imagined them as moving around. The on ones moved to the left, the off ones moved to the right. A global energy minimum now corresponds to the graph theory min-cut. This looks like the physical chemistry process of liquid-liquid phase separation. Molecules that like to stick to each other, clump together, minimizing the surface energy between the clumps. We looked into it, and indeed, phase separation looks like a neural network mathematically. And so now we're designing DNA molecules that phase separate according to this principle, as you can see in this confocal microscopy image. I think this nicely illustrates the power of finding the right perspective from which to look at things in a simple way that gets at their essence, and Shuki is a master of this. I am now transforming into Andre. Shuki has always asked important questions, and he has done it in a very playful manner. Shockingly, he has done this as much in biology as in computer science. He has said that biology is magical. I do not think he meant it in some sort of fairy tale way. Rather, he tried to express the apparent insufficiency of our language, scientific or not, to capture the wonders of biological phenomena. And what I remember the most, and keep repeating to my students, is that we need new languages to understand new things. To understand if biology is more than simple chemistry, or if cells really compute the way that we do. We still do not know the answers to these deep, though seemingly simple questions. Shuki worshipped Shannon. I remember his tribute when Shannon passed away. So, not surprisingly, he was the one who infected me with the idea that information can be one of the languages we should use to understand biology. This led to one of the most informative, pun intended, forays for me personally to describe biological signaling pathways as information transfer devices, measuring for the first time their information capacity in bits. This approach has proven incredibly influential and changed how we as a community think about biological signaling networks. But it also paves the way for new languages, such as quantifiable landscapes, biological regulation, particularly in epigenetic control. And more recently, the realization of deep similarities between computational neural networks and neuronal circuits and intracellular signaling. And as Shuki foresaw, new languages brought about new levels of understanding. Biology is still magical. The questions Shuki posed are still there, but he has also led us to some exciting answers. And I will always be inspired by the image of his smiling face as the magic of nature comes to focus again and again. Thank you, Shuki, says Andre. Our next presenter is Mark Riedel.
So thank you, Eric. I'm on the panel titled Biological Systems, but my PhD dissertation was on the topic of digital circuit design, specifically designing digital circuits with feedback. This was a topic that was controversial that I worked on for a few years and then worked on other topics and then came back to it. I spent many years doing my PhD, and Shuki was incredibly supportive throughout. On the topic of biological systems, it's already been discussed and alluded to in different ways, Shuki's views on biology are deep and profound and unique, even within the context of a place like Caltech, which has very unique views on what biological circuits are. It's come up again already in today's discussion. In fact, it's the title of the entire event, information. But Shuki focuses on, as an underlying concept, information in biological systems, and he also focuses heavily on probabilistic behavior, stochastic behavior. So these views definitely came out in discussions that I had with Shuki, and like Shuki, I remained throughout my career focused on circuits and theory, but I worked on biological systems. In fact, that's what I do primarily now. His views on biology definitely were percolating at the time, but the course IST4, which was discussed and will be discussed again, I think Shuki taught it later, after I left Caltech. Looking through the slides for IST4, you can see how bold he is in his statements. What is life? And life is a natural information system, and more specifically, how do you characterize all of life? How do you characterize molecular systems? How do you characterize the brain? All of these in the course on IST4 from the slides, an absolutely fascinating and deep view of what these are. What I think is true is that statements like life is an information system, other people have made those. The difference with Shuki is these grand statements are followed up with a tremendous body of theoretical research, and the theoretical research has deep mathematical underpinnings. So it's the broad vision coupled with the mathematical rigor that makes this work so transformative. Here is an example. Shuki observes, and again, I'm taking slides from the IST4 class here, you'll notice the fonts, that in biology, whether you're choosing to describe molecular systems or an entire biological system, such as a human, there's a huge amount of precision in what happens, how the body is formed, how the animal is formed. And yet, somehow, all of this is built from stochastic chemical reactions at its undercore. This is a very deep insight. Probabilistic behavior is a recurring theme in the analysis of biological systems and information systems. So some of the papers, I'll point to one here. I don't think Michael Gibson is here, right? Correct? But Shuki's most highly cited paper is a paper that he wrote with PhD student Michael Gibson on an algorithmic technique for simulating chemical reactions. And it again shows the depth here. This isn't information theory. This isn't a circuit design that I worked on. It isn't biology. It's algorithms. And it's algorithmic work that was profound in this paper. And then many other papers, some that we've seen. Dan is here. This is really a beautiful piece of work that Dan did with Lulu and Shuki on probabilistic analysis of circuits coupled with the implementation for DNA. And others, including work that I did after I left Caltech with Shuki and a student on stochasticity in biochemical systems, and the work that I hope we'll hear more about with Matt Cook and David Soloveichik on chemical reaction networks as a computational model. So I'll stop there, and I've managed to go over. The next speaker is Colin Chen.
Hi everyone, I'm Ho-Lin Chen, and part of my talk will be slides from Hongchao Zhou, who is a student in Shuki's group. And so I joined Shuki's group meeting back when I was a postdoc at Caltech from 2007 to 2011. So at that time, although I wasn't directly in Shuki's group, I was sort of attracted by all the interesting discussions and all the positive energies in Shuki's group. Shuki has the magical power of let everyone feeling welcomed in his group, no matter it's a student, postdoc, or just a visitor. He allows people from very different backgrounds to actually sit down and work together, discuss together. And so that generates a lot of very interesting collaborations. And one of the projects that came out from this discussion with work with me, Hongzhao, and Shuki was a work that we think about how to generate complex probabilities using simple molecular systems. So this work sort of started with the work by Dan, Lulu, and Shuki, that they designed molecular systems to create simple probabilities. But even before the molecular system actually works, Shuki already lead us to think about the power and limitations of these systems, that what kind of probabilities can be generated from very simple systems, and what kind of, how much resource is needed for that kind of models. So I think this is a very special way of looking at different molecular systems. We can analyze their power without actually going to all the molecular details. And so when I'm preparing this talk and looking at the slides that Hongzhao sent me, one of the things that we really want to emphasize that we'll remember is Shuki's smile. And of course his jokes. So Shuki has this very positive attitude, and he really enjoys everything. He's always smiling while he's like teaching, mentoring, research, everything. And so this attitude really influences everyone around him. So I remember when I was a postdoc here, I really had a difficult time finding faculty positions. So at some point, I even thought about just giving up and go find some other jobs. But Shuki is one of the key person who really encouraged me at that time. And so his encouragement and his positive attitude is something that really carries me through several years of job search and really lead me to who I am right now. So thank you, Shuki. Our next speaker is Damien Woods.
Hi, everyone. So I was here for about 7 years, between 2009 and 2010. I arrived as a theoretical computer scientist, and I was a senior postdoc. So I already thought I knew a little bit about theory. But I decided to attend Shuki's course, IST4, anyway, and my gift to you is that I will just talk about that today. That's all. So one thing about the course, besides the Comic Sans and bright yellow backgrounds and all sorts of crazy things, was that there would be a lot of questions, and he would stare you in the face, and somehow he could stare at everybody in the room at the same time, and he would ask you all to answer this. And sometimes the questions were mathematical, and maybe you'd have some hope if they were easy, and sometimes they were weird. He didn't ask this, but I'm asking this today because this is the picture I remember most from the lectures. And so it's a plot. There's some things and there's some milliseconds. What's going on? Does anyone have any idea who it is, who is not Shuki, and who did not TA the course? No, okay. So Shuki, what is the plot? Do you remember? Yes. So when we look at objects, if there's 1, 2, or 3 of them, we can recognize that number immediately. And it takes about the same time to recognize each of those numbers, like we see the color red or blue or something if you're not colorblind. Whereas as the numbers get bigger, you seem to take more time to figure out what's going on. And so Shuki explained this is why we need number systems. And this was one of the many starts of IST4. There are actually several starts to that course. There are several ends, and there's a lot of middles too. But what he did was, he said, there's going to be 2 ideas that I mentioned in my slides. Idea 1 is to learn from the old masters. That sounds like a very heavy term, and that is not the term I come to learn these people by on this slide. There's an awful lot of people here. We have, some people, we don't have their faces from 20,000 years ago who put some notches on some bones, and even there was a bone from 30,000 years ago with 29 notches on it that we figured maybe it was to measure a lunar cycle. We had the Babylonians, they wrote in base 60, so Shuki said they knew everything, primarily for that reason, but I think he had other reasons too. Later we learned about algorithmy, about people from all over the world and all over history. 3 of the major heroes, I would say, if I had to pick some, were Leibniz. We had some Leibniz cookies. George Boole, and I spent some time at university where George Boole was a professor of mathematics. And of course, Claude Shannon, everyone knows, is Shuki's great hero. And so why was he telling us all this? Was it a history lesson? No, it was actually about biology. He was trying to tell us that what we need to do if we're going to figure out how does biology compute, how does it work, how does it manage information, well, let's take a look at what all these people do. Let's understand their stories and remember them as people and try and figure out where they were, why did they come up with their idea, and then how did that bring our field forward. And so maybe we could apply those ideas to biology. Although the second idea was, and inspired by all those people, was that because biology is so complicated, let's do synthesis instead of analysis, right? You look at a cell, you look at an organism, you look at me, you know, everything's very complicated, so let's try and synthesize some things. And he did say about this slide, this picture right here, if you see me in 20 years, remind me of this slide. It's been 16 years since you give this slide. And what are these smiley faces? It turns out they're a chemical reaction network. They're interacting together, and they're going to do something like build this worm that has a fixed number of cells. So how could you have a noisy bunch of interacting molecules that could do a deterministic thing like build that? So he wanted us to try and figure that out. On the right-hand side of the slide, I just threw a few quotes. Of course, the most important thing in research are the questions. So I also am one of these people who believe strongly in coming up with good questions, and I always find it hard to teach students how to do that, and I remember Shuki's advice on some of these things. But my favorite quote is this: if you're trying to sleep and someone wakes you up, and what's their problem? Their problem is they can't prove something and it's really hard. So what you should do is just wake up, shout, "Induction!" and go back to sleep. Thank you very much.
Wonderful. We have 8 minutes and 45 seconds for discussions, so I'm looking forward to that. I hope you've been planning what you'd like to ask. To accompany our chat, I'm showing a painting by Anne Urpino that I commissioned as part of a series that honors students, colleagues, and co-authors who have made my life richer. This one was done for Shuki. It tries to reflect the idea that to get at the mathematical essence of something, you need to make an abstraction that throws away the inessential and leaves you with something that actually doesn't look so much like the real thing, but is a powerful conduit for deep insight. Shuki is a master at that. Okay, let's hear from the audience. They're very shy. I will just sit over here and wait. I'm looking at all of you at once.
Can we ask Shuki questions?
No. So Shuki, sorry,
use your microphone.
I think it's on. Sorry. Yeah. So Shuki used to make a lot of bad jokes in IST4, and one of them was about his own accent. He said, "My accent will never change." And then, he would call people out in the lecture, and for some reason I actually, on the plane over here, I watched the entirety of IST4 from 2010. I kept the recordings. And in that, you make jokes about your accent, Shuki, and then at some point you called me out and you say, "Someday will you teach me an Irish accent?" And no, I failed in that regard, I think.
Ellen.
Okay, I'm a latecomer, so I apologize, but I am a biologist, and I'm very curious what kind of answers you guys came out with for explaining how biology is set up to make discrete decisions that are very clean out of extremely stochastic inputs and underlying mechanisms. And we know lots of details about this, but it would be really great to hear your own perspectives on what the essential features are that make this possible from a mathematician's point of view.
Damien, you go.
Do you want me to ask again? It's just, what is your own perspective of the answer of the question of how biology makes these clean, discrete decisions out of such stochastic, reversible chemical processes?
So if I was going to say something quickly, I'd say there's a lot of clever error correction going on, and so there are you can with stochastic processes, get the right answer. So if you've got a biased coin, and you want to get an unbiased bit out of it, there's a very simple technique where you keep throwing the coin twice in a row, and eventually you will get an unbiased heads or tails. So I would say, learning from Shuki's lectures, one thing I would say is you can employ lots of little simple tricks like that, and I think biology does some of those things just at a high level.
Or maybe let's see if I can articulate this, that you can have, there we go, better? Yeah, that you can have precision with randomness. Even if you have a tremendous amount of randomness, you can have a precise statistical measure that emerges from randomness. And I think Shuki articulated this better, but complexity and precision in, let's say, developmental biology probably arise from a deep mechanism like that. All the uncertainty, molecules bouncing around, it seems like chaos, and there's a huge gulf then from physics up to that level of biology, but then to emerge from that level of biology to complete functioning organism is probably a deep mathematical principle involved, which is precision in the statistics, not in the individual outcomes.
Until there's another question, let me follow up a little bit more. So developmental biology isn't just giving certain cell types, it's also giving very precise kinetics and very precise spatial connections between them. So when that little worm that you were talking about develops very, very, very fast, it doesn't flutter around in some kind of space for a long time. So when we look at the 300 to 500 genes that are most important for this kind of process to work, I'm very curious what you would say are the principles we should be looking for that would explain which ones are likely to be the most important for the correctness of these decisions in time as well as in outcome.
So let me maybe offer two perspectives. So we've heard a lot about how Shuki introduced neural networks, and not just as a artifact that you engineer, but as a concept, a way of looking at things. And so maybe we can take two kinds of ideas from that. One is that when you have individual sort of circuits that are based on a small number of components, small number of inputs, it's really easy for them to do the wrong thing. When you have a neural network that has many inputs, actually, neural networks are very robust to noise. It's sort of an intrinsic aspect of this high-dimensional operation. So maybe that goes some way towards helping think about biology also. A second thing about neural networks is, well, there's maybe two classes of neural networks that people study a lot. One are deterministic neural networks that sort of, like in Shuki's complexity theory, they exactly compute some Boolean function. Maybe just do that more efficiently. But another kind of neural network that has actually made way for the recent advances over the last decade have been probabilistic neural networks. And these are networks that actually aren't meant to represent sort of discrete input-output relationships. They're actually meant to represent a probability distribution. They're actually, the stochasticness isn't a mistake, an error. The probability that you have of the probability of this thing versus the probability of that thing is how likely it is, and that's the model, that's the information it's trying to represent, in a sort of Shannon sense maybe even. And so maybe the other part of the question, although maybe this doesn't address C. elegans, is that stochasticity is sometimes exactly what you want and need rather than something you're trying to get rid of.
I think I have a question sort of building on that. I think with respect to biological neural networks, they are these computing systems that have also these inherent stochastic spiking natures to them. And this probably isn't a definite question to ask, but from your perspective, do you think that the power of the biological neural networks comes from its inherent stochasticity?
Maybe we should vote on that. I think yes. And I think stochasticity also gets at another aspect of biology, that it's easier energetically to make a stochastic decision, to be half times right, half times wrong. Actually, that takes no energy. To drive something to be very perfect actually takes more energy. And so if the right answer, the right way of answering some kind of decision is to get the probabilities right, then the lowest energy way of doing it is to not force things to be discrete, not force things to be definitely 0 or 1, but to let them bounce around just the right amount. I think that reflects with what Mark was saying about generating stochasticity.
Ambitious question. Ellen brought up the notion that at the level of the molecules, everything is reversible. And of course, the arrow of time in physics is set by increase of entropy. And in a sense, biology is a system that is incredibly good at reversing that, at organizing molecules in such a way that entropy, in fact, at least locally, decreases. Have you come to any insights into what sort of mechanisms, what sort of general principles are at play at making that possible in such a successful way in biology, but not in other realms?
Anyone want to take that?
I guess I could just say one comment, maybe. Yeah, biology seems to be really good at striking that mix of. So we have our computers, which are extremely driven, by fuel, and they get hot and they consume a lot of energy, and it's very, deterministic. And biology seems to do this mix of consuming a lot of fuel. I wasn't able to get up here today until I had a little pastry earlier, but I didn't need that much, There are a lot of reversible processes within biology happening all the time, like at the local level, and maybe we understand those a little bit less. And I'm sort of really curious about that, like how much can we get away with if our chips, for me it's DNA chips, right? But if our chips were essentially working at thermal equilibrium or very close to it, or absolute minimal sort of driving forces. So I think there's a bit of that going on at an abstract level.
So we're already 2 minutes out of time, but I'd like to give the last word to Andre, who I'm going to paraphrase what I'm sure he would have wanted to say, which is that these kinds of questions about information and neural networks and stochasticity and decision making all reflect the need to help develop a new language for thinking about information processing in biology.